Plant tissue culture is a revolutionary technique enabling the rapid propagation of desirable plant varieties. It involves growing plantlets in a controlled, sterile environment, often referred to as in vitro. While this method offers numerous advantages, the transition from the in vitro environment to the outside world can be challenging for these delicate plantlets. This process, known as acclimatization, is crucial for the plantlet's survival and future growth. Acclimatization involves gradually exposing the plantlets to the natural environment, allowing them to adapt to fluctuating humidity, temperature, and light conditions. This transition must be carefully managed to ensure the plantlets thrive. This essay will guide you through the critical steps involved in successfully acclimatizing hibiscus plantlets, focusing on a step-by-step approach that emphasizes clarity and simplicity. By understanding and implementing these steps, you can significantly improve the success rate of your plant tissue culture endeavors and ensure the healthy development of your hibiscus plantlets. Each stage of this process plays a vital role in preparing the plantlets for the challenges of growing in a natural environment. Before we proceed into the acclimatization process, it is important to understand the environment in which the plantlets are initially grown. In vitro rooting is the first step in this journey. Plantlets are typically grown in a specialized medium within sealed containers. This medium provides all the necessary nutrients and hormones required for growth. This controlled environment ensures optimal conditions for root development. The containers are kept in a propagation room with carefully regulated temperature, humidity, and light. The high humidity levels in this controlled environment prevent the plantlets from drying out, a critical factor as they develop a robust root system. The propagation room is the heart of the in vitro rooting process. It is designed to provide the ideal environment for the delicate plantlets to develop strong roots before they are exposed to the outside world. Temperature, humidity, and light are meticulously controlled to mimic the optimal growing conditions for the specific plant species. The propagation room is typically maintained at a constant temperature of 20 degrees Celsius, both day and night. This consistent temperature provides stability and prevents temperature fluctuations that could stress the young plantlets. Humidity is kept exceptionally high, around 96%, to minimize water loss from the plantlet's leaves. Lighting is another crucial aspect of the propagation room. High-pressure sodium lamps are commonly used to provide a consistent and intense light source. The lamps are set on a timer to provide 16 hours of light followed by 8 hours of darkness, simulating a natural day-night cycle. Section 4. Preparing the peat a sterile foundation. Once the plantlets have developed a healthy root system in the controlled environment of the propagation room, they are ready for the next stage, transplanting into individual pots. Peat, a naturally occurring fibrous material, is commonly used as a growing medium for acclimatizing plantlets. Before being used, the peat must undergo a sterilization process to eliminate any potential pathogens that could harm the young plants. This is typically achieved by autoclaving the peat at a high temperature and pressure. The peat is placed in an autoclave and subjected to a temperature of 121 degrees Celsius or 249.8 degrees Fahrenheit and a pressure of 15 pounds per square inch for a duration of 20 minutes. This intense heat and pressure effectively kill off any fungi, bacteria, or other microorganisms present in the peat. Sterilizing the peat ensures that the plantlets are transferred to a clean and disease-free environment, reducing the risk of infections that could jeopardize their survival during this vulnerable stage. Section 5. Transplanting to pots. The gradual transition. After approximately 15 days in the propagation room, the plantlets, now with well-developed roots, are ready to be transplanted from the in vitro environment to individual pots. This transition marks a significant step in the acclimatization process, as the plantlets are gradually exposed to a less controlled environment. Carefully remove the plantlets from their sterile containers, handling them gently to avoid damage to the delicate roots. The sterilized peat is lightly moistened to provide adequate moisture for the plantlets. Each plantlet is then carefully placed in a small plastic pot filled with the prepared peat. The pots are then moved to a greenhouse where the humidity is still relatively high but lower than the propagation room, typically around 70%. This gradual reduction in humidity helps the plantlets to acclimatize to a less humid environment, preparing them for the conditions they will eventually experience outdoors. Section 6. Greenhouse Care Nurturing Growth. The greenhouse provides a controlled environment that acts as a stepping stone between the propagation room and the open field. While the greenhouse offers some protection from the elements, it also exposes the plantlets to more natural fluctuations in temperature and humidity. This exposure is crucial for their continued acclimatization and overall development. Inside the greenhouse, the temperature is maintained at 22 degrees Celsius during the day and 20 degrees at night. This slight drop in temperature at night is beneficial for the plantlet's growth and hardiness. The humidity is maintained at around 70%, significantly lower than the propagation room but still higher than typical outdoor conditions. To support their growth, the plantlets are irrigated daily with a standard fertilizer solution. This provides them with the essential nutrients needed for healthy development. The gradual decrease in humidity and the provision of nutrients through fertilization encourage the plantlets to develop stronger, more resilient leaves and stems.